Thank you very much. Welcome back, everybody. My name is Carsten Skarke, and I'm from the University of Pennsylvania. Can everybody understand me loud and clearly? Good, yeah. So 6 years ago, I started to develop an interest in biorhythms and chronobiology. And we came up with that term chronobiome to label or to talk about time-specific phenotypes, so both in health and in disease. And that coincided, this increasing risk, because I realized that we have an increased amount of wearables, remote sensors at our hands where we can measure over time at high and highest resolution, right? And if you're interested in biorhythms, what better can there be than a data stream at high resolution, right? And so to be a little more provocative, the vision is sensor to molecule, so meaning or asking the question, how much can we use the wearable, the smartphone, as a proxy or a window to look into what is going on a molecular level in relation to health and disease and the trajectory, how health changes to disease phenotype over time. And so we take the specific look and interest in the variability occurring over the course of 24 hours. And so that's depicted here, right? So for many measurements, depending on the time, you get back a different level of abundance or whatever your outcome of measurement is, right? And so to illustrate the clinical relevance for that, here we look at a blood pressure curve over 24 hours. On the left side are the nocturnal measurements, so during the night, and on the right are during the day or wake times. And in green are young volunteers, and in orange are old volunteers. And so immediately you see that there is an age difference, right? So overall the old participants have, on average, higher blood pressure measurements. But then you see also this diurnal phenotype, right? You see this dip at night, and that's to be expected as part of a healthy phenotype. And then, of course, higher blood pressure levels throughout the day. And the interesting aspect of this diurnal phenotype is that it becomes, with the supporting research, it becomes clinically actionable. So if we deal with a patient where this curve is elevated consistently, it's a case of hypertension, and we have many antihypertensives in the clinical portfolio to address that. But then there is another time-specific disease phenotype where the dip at night disappears. So basically you do measurements, you discern, oh, that's an hypertensive patient, and then you look again and it's basically more or less a flat line, meaning the lowering of the blood pressure during the night has disappeared. And so that's a time-integrated way to look at such a phenotype. And clinically we know that patients with this disappearance of that nocturnal dip are at much higher risk of cardiovascular disease down the line. And then the question is, well, how do you make that actionable? And there are clinical trials out there which give an antihypertensive in the morning and in the evening. Both approaches lower the blood pressure equally, but the same medication administered in the evening has the effect that it reconstitutes the nocturnal dipping phenotype. And so with this time-specific administration of medication, you reconstitute this more healthy phenotype, and you would expect that down the line, you would decrease the cardiovascular risk in this hypertensive patient. So that's the clinical relevance. Why do we see diurnal variations over the course of 24 hours? And that is basically hard-coded into each of our cells. And that's the molecular circadian clock. And it's basically a feedback loop where certain proteins or gene transcripts increase the transcription of a different set of proteins, and these circle back and decrease the transcription temporally of these initial genes. And so that is over 24 hours and basically constitutes the circadian diurnal aspect of many physiological, behavioral, and molecular phenomena. And this core clock is tied into clock control genes, and so blood pressure regulation is tied into these core clock genes, and so that is basically the mechanism. And we have a central clock in the brain and peripheral clocks in the organs, so it's widespread. There's redundancy, so you see these pairs here in the system. It's highly conserved across species. It enables to compartmentalize processes by time and not only by space. And it's this anticipatory system to ensure biological fitness. To go back to that blood pressure example, more or less 1 hour before we get up, our blood pressure starts to rise in anticipation of that we need to go from horizontal to vertical. That's the biological meaning there. And so our internal circadian clock is running more or less at 24 hours. If we sample the room, the majority would take a little longer, 24 hours and 10 minutes, a few less. And so then this gets entrained to exactly 24 hours by light as an environmental cue, but also by physical activity, social time, meals, etc., because we're very much embedded. And so then these cues synchronize basically these 24-hour feedback loops, right? And so that's the molecular mechanism tied into our environment. And jet lag is an example to disrupt or misalign or deconsolidate these circadian or diurnal rhythms. Devices, when we try to go to sleep, blue light exposure, shift work, and many diseases are associated with circadian disruption. And for the last example, we took a look at the UK Biobank and looked at the biorhythms specifically in the temperature trace collected in that Exivity device, which was given out to around 100,000 participants in the UK Biobank. And we were interested in what do we see in these temperature curves? Is it completely contaminated by ambient temperature, or do we see a biological signal, etc.? And the UK Biobank is wonderfully set up that it allows us to pick from the cohort of actigraphy wearing participants, the ones at that time without a code of, without a phi code, so without an association or no notation of a disease phenotype. And then ask the question, well, among these increasing diagnostics and reported death, how does that relate to the initial temperature curve? And to look at that temperature curve, it's peripheral temperature, so the core body temperature is directly controlled by the circadian system. And at night, similar to the blood pressure, you see this nocturnal dip in body core temperature. And in order to to cool down your body core, your periphery opens up the vessels to to dissipate heat. And so this is what we see here during the night hours. The wrist temperature increases by 2 to 3 centigrades. And then at morning, you have this typical sharp drop. And then you have this shoulder, and that mirrors exactly what has been reported in much more controlled studies in the literature. And so as a kind of a sanity check, here we look on the left, we look at nap during the day. And you would think that the more naps you report, the more disrupted is your wake-sleep rhythms, right? And so here we see that in blue, the amplitude is highest in people never reporting a nap. And people who usually nap is in green, and you see that the amplitude between night and day oscillations in that temperature curve is decreased, right? And so this is according to expectation. And similarly here on the right, there's a chronotype questionnaire, morning versus evening person, and this behaves exactly the same, that the evening person in red here is right shifted compared to the blue morning person, right? And so that's exactly what we would expect from a biological data trace, right? Of course, there's probably contamination by ambient temperature fluctuations, but this is the sanity check that we do see a biological signal. And so in patients, to take an example, in patients with diabetes mellitus, when we compare these, and these are in the orange curve here, when we compare these patients to controls, so without any Phi code diagnosis in the health records, there's a significant decrease in that temperature amplitude. When we plot this as a function, so this temperature amplitude, when we plot that as a function of the disease incidence, this relationship surfaces where the higher your temperature amplitude, so the higher your fluctuations, the lower the incidence among those people or participants for a diagnosis or future diagnosis of diabetes mellitus. On the other end, if you have almost a flat line, so very low amplitude at 1, for example, here, you have a much higher incidence in that cohort of diabetes mellitus. And when we go through all the Phi codes, right, and run this as a Cox proportional hazards model, then this 2 standard deviation decreased amplitude in these temperature fluctuations are predictive for diabetes, more than 50%, 50%, hypertension, 25%, and so on. And interestingly, like all of these, you look up the literature and you will find many papers suggesting that that there is an association with circadian clock disruptions. Looking into all-cause mortality, that is also, so we saw that it associates with a higher risk for future disease diagnosis, so you would, from that, deduct or infer that the mortality would also be higher, and that's exactly what we see here. Currently, there are about 3,000 reported deaths in the UK Biobank. So that is to highlight the power of, in this case, just 1 week of peripheral monitoring on your wrist, how powerful that can be for diagnostic capabilities and making this information clinically actionable. And so to go from this population level into deep phenotyping, we came up a couple of years with the study paradigm of the human chronobiome. And basically, what we combined was a suite of wearables, smartphone apps, etc., and combined that with, for that pilot study, just morning and evening biosampling, so blood, urine, saliva, etc. And we did that study in the wild. So we invited the noisiness of our lives into our data set with the question, can we, under these circumstances, detect time-specific differences in the wearable data and in the multiomic data? And of course, in the wearable data, it's very clear that activity, blood pressure, heart rate, etc., there's a strong diurnal component. But the big question was, well, in the multiomic data, in that noisy approach, can we detect time-specific elements? And so here we just decomposed variability. We look down here what time of day contributes to the variability, and this depicts the inter-subject differences, right? And so below the line of identity, these are elements across the metabolome, microbiome, proteome, which have a strong time-specific characteristic. And so with that limited data set, just morning versus evening comparison, we asked the question, well, if we do a network analysis, would we see differences in enrichment in these networks, right? And in this approach, we saw that cancer and chronic inflammatory disorders show a time of day specific enrichment. And that caters again to what is known in the literature. And so we took this paradigm of this pilot chronobiome and now scaled it up to, and I wanted to say, so we started with the healthy population in the interest to create and establish a reference before we go into diseased populations with this kind of approach, to know exactly when we see disrupted features in disease, how does that compare to the reference, right? Makes sense. And so here we scaled it up to look at age-dependent effects on oscillatory networks. And our hypothesis is that deconsolidation of oscillatory functional networks contributes to age-related disease expression, right? Over time, as you age, your blood pressure increases, and that increases your risk for cardiovascular diseases downstream. And we're interested in that. And scaling up meant, so we went from 2.2 million data points collected for our pilot to now 75 million data points in our 20 subjects. And we have a clear separation by age. So on X is the chronological age, so our young and our old cohort. And then we took the methylation data we collected. And you can determine the biological age by submitting selected CPG sites and their abundances to the Horvath clock estimator. And then you get, for each time point, and we have 9 different time points over 48 hours, you get an estimator, right? And notice how variable your biological age is over time, right? So we look at a differential of, in many cases, more than a decade, which of course brings up the question, am I younger in the morning or in the evening? Should I plan my activities accordingly, right? So that was an interesting finding. In terms of wearables, we use those, in this case, to functionally separate by age. So grip strength, for example, response time, audition. You clearly see the separation by age. Green is young, orange is old. These functions, there is a known diurnal variation existent. We saw it less pronounced in our data set. I think it's a numbers game. We just compared 10 and 10. Usually these reference studies are done in hundreds or thousands of patients, where you clearly see a diurnal phenotype. And then just in terms of physical activity, they're very similar, but here you see that the old are phase advanced, so they're active more in the morning hours, which is expected. Light exposure, also similar, but the young tend to get a little more. Kind of sanity checks, and then also EKG data. And we have a way through Kubios software out of Finland to look at indices which indicate the activity of the sympathetic and the parasympathetic nervous system, right? And so we do see strong diurnal phenotypes which look age appropriate. So in the old, overall, the sympathetic nervous system seems to be more activated than in the young. Still this diurnal phenotype, and then the parasympathetic down here, it's flipped as we would expect. We followed also with glucose levels, and just to make the case that glucose levels are pretty normal, but you see that trend that they are slightly higher overall in the old. And here just two wonderful diurnal profiles in young and old of how your glucose measurements fluctuate over the course of 24 hours, right? And so that now makes the case how important it is to look at an unbiased omics level at these diurnal fluctuations, right? And so here, this is data on the top, the young and the old on the bottom, and these heat maps represent, so one column are our 10 young in this case, measured at 8 a.m. in the morning. The next column is our 10 young measured at 12 p.m., 4 p.m., etc. And then the each row on the left here is one transcript, and you see that like the top more or less 50%, you see that the abundances, they fluctuate as you go through the 24 hours, right? And they are ranked by the sinusoidal fit. So on the top, that's like the most sinusoidal transcript. And so we look at the methylome, the proteome, or two flavors at the proteome, the metabolome, and overall there are many features which fluctuate, so which have this diurnal variability both in young and old. And this is about 1/3 of the ohms we are working with. So it's a very comprehensive approach. And this looks now at, so another sanity check. So we did see an enrichment of features in the evening for both the transcriptome and the proteome, right? So the young are on the left here in the transcriptome, the old on the right, and you see this more denser cluster on the high in the evening annotation here, right? And so this is in the old, and then the proteome here, it's similar. So we have a congruence across, a transomic congruence, right, of this signal in terms of a sanity check. This is now an example from the saliva metagenomics, where we collected in equal at the same time points. And the blue are the old and the young are the orange here. And you see both are cyclic, but the characteristics of that curve are overall different, right? So in terms of like the phase, so the peak are different, the amplitudes are different. And so this was very comforting to see that also in a pathway level. So if we move from like single features to a pathway, that these fluctuations are also evident on the pathway level. So Anthony Marcella is up there, he adopted a platform called Mixomics, which allows us to integrate all these ohms. When we, so this is one of the diagnostic plots, and very interestingly and comforting again is that our both age groups, when we create these oscillatory transomic networks, they clearly separate. So the orange are young on the right and the blue are on the left. So that is, again, another sanity check. And then this platform lets us allow to hone in on features which are highly correlated, but retain this discriminatory ability to discern young versus old, right? And so here in the circus plot, we get between all the ohms, and here we have also the phenome, so heart rate, so heart rate variability measurements, PM 2.5, because we looked into air quality, also blood pressure, etc. So here now we have these cross-correlation networks where we then can get more or look deeper into the molecular mechanism and how they, what kind of story they tell. And this is ongoing work, so this is very fresh still. But one of the main features which consistently pops up as the top connected feature is FGF21. It's a sensitizer and controls metabolism, etc. So it turns out it has a high abundance in the old, and it fluctuates or is sinusoidal in both. And so this reflects exactly what we see in the literature. And so now this is, for example, one a start where we dig deeper into how, on the molecular level, what kind of stories this tells us, right? So to conclude, in the UK Biobank, risk temperature traces are predictive of future diseases in the human chronobiome, so we do see unique characterization of oscillatory physiology and biochemistry in healthy individuals. There are striking molecular oscillations in the basal state with healthy aging. And this time-integrated transomic modeling allows us to look now at top candidates to drill deeper, right? And this is one bioinformatic approach. There are, of course, others, and we'll diversify these modeling approaches. And next step, next year, we'll go into diseased populations to really build on that and to understand, well, how on an oscillatory level, how do disease patients differ from healthy participants? And so this is my prompt. Well, how do smartphones-based data fit into that? So we also collected their data, their behavioral data with BWI. We have that data set sitting, so we want to parse that and come up with interpretations how that relate to the multi-omic data set. And here just, we also collected the hungry caterpillar voice samples from our individuals, and we had them analyzed for emotional valence, for sadness, joy, etc., to explore an unbiased way to look at emotions in all of that. And just the last one here is, so I mentioned with the blood pressure that there are therapeutic opportunities to modify in a time-specific way. And this is a paper in mice, which basically says that 2/3 of the top-selling prescription drugs oscillate in mice, and most likely in humans too. So there is a component of adjusting in a time-refining way the therapeutic approach. So lots of people contribute to that, thanks to my mentor and long-term colleague and collaborator, Garrett Fitzgerald. And then over the last year or 1.5 years, it's basically data crunching with Anthony Nicholas Lyons, Tom Brooks, Jonathan O'Brien, Eugene Malamud. And lastly, cardiology, it took 13 Nobel Prizes to create that field. Circadian clocks just got the one a couple of years ago. Go for it. Thank you.